Hello students. So we are going to today discuss about mineral nutrition in plants. So what is mineral nutrition? To define mineral nutrition, it is a study of how plants obtain, distribute, metabolize and utilize mineral nutrients. As shown in the picture, you can see carbon, oxygen and hydrogen. They are essentially obtained from the atmosphere in the form of carbon dioxide and water vapor by the plant. Some of the colored dots which you see on the soil or the underground region of the plant, this is an inorganic substance which is obtained from soil, which are called as mineral nutrients or mineral elements. Now these elements, they are transported across various organs for specific functions like germination, growth, or to fight against pathogen, diseases and also for reproduction. Now why should we study about mineral nutrition? Now we know that plants are used by us for various applications. The major application of it is in food. So in agricultural practices, in order to obtain maximum yield of crops, it will be essential to match the crop's mineral elements supply and demand throughout its growth period, so that we can achieve maximum yield and prevent any kind of nutrient deficiency or its toxicity which can hamper the growth of the plant. So in this chapter, we are going to identify mineral elements which are essential for the growth of the plant. We are going to talk about the criteria to classify them as essential mineral elements. Further, the essential elements are divided into macro and micronutrients. We are going to study about the absorption and their role in plants. Moving forward, we will also discuss about the deficiency symptoms of these essential elements in plants and what happens when they are provided in excess, causing toxicity in plants. So majorly we will be focusing on toxicity of micronutrients, then mechanism behind the absorption of these essential elements, and in the end we will discuss briefly about the metabolism of nitrogen in plants, including the different ways in which nitrogen is assimilated in the plants. Now coming on to the question, how does one identify whether a mineral element is essential for the growth of the plant? Now in 1860, a prominent German botanist, he demonstrated for the first time that the plants could be grown to maturity in a defined nutrient solution with the complete absence of soil. So this technique of growing plants in a nutrient solution is known as hydroponics, where now it is being commercially utilized for the cultivation of various crop varieties, food crop varieties essentially. So if you see the picture on the slide, you can see how tomatoes and lettuce is being grown using hydroponic cultures, and the schematic shown on the slide shows the arrangement of the stacks in which the plants are potted, such that the roots are exposed to the nutrient solution and the nutrient solution is recirculated to all these stacks of plants using a pump, and the solution in turn is aerated to make oxygen available to the plants. Hydroponic system works by allowing minute control over environmental conditions like temperature and pH balance and maximum exposure to nutrients and water. Now, you must be wondering why there is a stone kept in the lower chamber of the schematic. The purpose of this air stone is to gradually diffuse air into the tank, eliminating the noise and the large bubbles of conventional air bubbling or air filtration systems. Mineral elements are further classified as essential and non-essential elements. So, what is the criteria which makes a mineral element as an essential mineral element? So, the criteria of essentiality includes these essential mineral nutrients. They are absolutely necessary for growth and reproduction. Essential mineral nutrients are specific and non-repressible by other elements, and they are directly involved in the plant metabolism. So, the essential elements, they are required for the plant effectively to complete their life cycle. As shown in the picture on the slide, the life cycle of a plant is being demonstrated, beginning from the seed, the young plant, going to a maturity, then the flowering, fruiting, and then again coming back to the seeding. So, some of the essential elements, they are circled in red here, like carbon, oxygen, iron, nitrogen, hydrogen, and phosphorus. While the non-essential elements, they are beneficial for the higher plant metabolism. For example, cobalt, sodium, selenium, and silicon. Higher plants mean the plants which have a well defined vascular system, including the xylem and the phloem, and higher plant metabolism here. So, just to give you an example how some of these non-essential elements, they are involved in higher plant metabolism. Selenium has been reported to help the plant cope with the stress by the enhancement of the activity of antioxidant enzymes. The essential elements are further classified as macronutrients and micronutrients. These are based on the quantitative requirements of a plant. Like when they are required, the elements, the essential elements which are required in the amounts greater than equal to 10 millimolar per kilogram of the dry plant matter, then they are classified as macronutrients. So, the elements shown here in the purple color circles, these are the macronutrients, which include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulphur, potassium, calcium, and magnesium. The essential elements which are required in quantities less than 10 millimolars per kilogram of the dry plant matter, they are classified as micronutrients. So, those which are circled here in orange. These include iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel. So, in total there are 17 essential elements which are required for the plant growth and development. Now let us see how these macronutrients, they are absorbed and what is their role in the plant. Talking about the three basic elements, carbon, hydrogen, oxygen, we all know that they are absorbed in the form of carbon dioxide and water vapor by the plant. You can see them, all the three elements, as a part of various biomolecules in the plant, including your carbohydrates, shown here some of the examples like glucose, fructose, then proteins, where you can see carbon, hydrogen and oxygen as their structural components, and in fatty acids. Let us come on to nitrogen. Nitrogen is absorbed by the plant in its ionic form of including nitrate ions, nitrite ions and ammonium ions. Nitrogen, once assimilated, it becomes a part of the protein molecules. You can see nitrogen circled in purple, shown for an example protein molecule. Then nitrogen is also a component of nucleic acids like adenine and guanine, shown here. The nitrogen part, to show you, has been circled in purple. In some of the vitamins like thiamine, nitrogen is a part of its structure. Then in one of the other biomolecules, the chlorophyll, which we know the colored pigment in the plants. So, in the chlorophyll molecule, nitrogen is also included in its structure. Then nitrogen is also a part of the structure of indole 3 acetic acid, which is a plant hormone and auxin required by the plant for the cell elongation. So, there is production of auxin. So, auxin is transported to the tip of the stems where it gets accumulated in the presence of sunlight on the darker regions. It gets accumulated, causing cell elongation. Coming to phosphorus. Now, phosphorus is absorbed in the plant in the form of dihydrogen phosphate ion and hydrogen phosphate ion. Phosphorus is included in biomolecules in the form of phosphate groups like the structure of phospholipids shown here in the cellular membranes. So, if you see, the phosphate group has been circled in purple here. So, phosphorus is also included in the biochemical reactions involving addition of phosphate groups. An example is shown here where hexokinase or glucokinase is involved in the addition of phosphate group to D-glucose molecule, converting it into D-glucose 6-phosphate. Phosphorus is also a part of phosphate groups in the phosphodiester bonds of nucleic acid, shown here again in circles, the phosphate diester bonds. Now, how is sulphur absorbed in the plants and what is the role of sulphur? Sulphur is absorbed in the form of sulphate ions. There are sulphur containing amino acids like cysteine and methionine. It is also included in the as a structural component of coenzymes like the coenzyme A, which has a role to play in citric acid cycle and fatty acid biosynthesis. It is also included in some of the vitamin molecules structure like biotin. Talking about potassium, potassium is again absorbed in the form of potassium ion. Potassium ion along with water movement is responsible for the opening and closure of stomata as shown in the picture here. It facilitates the transportation of sugar, nutrients and water. Therefore, it is required in abundant quantity for meristematic tissues, buds, leaves and root tips. It also helps in maintaining the ion and cation balance with the help of other ions like chloride ions, which helps in cell osmoregulation and maintaining the turgor. It is also an activator of certain enzymes which are involved in the biosynthesis of proteins and starch. So, now talking about calcium absorption and role in plants. It is absorbed again in its ionic form. Calcium in the form of calcium pectate, it facilitates the binding of adjacent plant cells. It also acts as secondary messenger, a signaling molecule which activates the cellular response towards stress conditions like drought, salinity. So, calcium ion is also called as a secondary messenger which regulates cellular activity. It is also included in the formation of mitotic spindle. Another element, magnesium, it is absorbed in the form of magnesium ion shown in circle here. Magnesium is also included as an activator of enzymes which are required in cellular respiration, synthesis of DNA and RNA. Magnesium is also a part of the chlorophyll molecule and it is also included for the maintenance of the structure of the ribosome, the large and the subunit fitting, which are sites for protein synthesis. Now, let us see how micronutrients are absorbed in the plant and what is the role of these different micronutrients. Starting with iron, how is iron absorbed and its role in the plants? Iron is absorbed in the form of ferrous ions. It is used in the formation of chlorophyll. It acts as an activator of catalase enzyme which protects the plant from oxidative stress due to hydrogen peroxide. So, ferredoxins, they are iron sulphur proteins, and cytochromes, these are redox active proteins which contain a heme with a central iron atom at its core, which acts as a cofactor that mediates electron transfer by changing the oxidation state of the ion from ferrous to ferric. Starting with iron, some idea on manganese. Manganese again, it is absorbed in its ionic form. It is used as an activator for certain enzymes in the Krebs cycle like succinate dehydrogenase, malate dehydrogenase. It is also included as an activator for nitrogen reductase, which is required during nitrogen metabolism. Manganese is also involved in the light reactions where it facilitates the photolysis of water molecules and provides energy for photosynthesis. How is zinc important for the plant? It is absorbed in its ionic form. Zinc is included as an activator for superoxide dismutase, which prevents oxidative degradation of hormones like auxins. It is also included as an activator for carbonic anhydrase. Carbonic anhydrase is involved in biochemical reactions which are responsible for pH regulation, CO2 transfer and stomatal closure. How is copper useful for the plant? It is absorbed in its ionic form, cuprous ions. You must have observed brown pigmentation in ripe bananas. So, that brown pigmentation is due to the activation of the biochemical reaction caused by polyphenol oxidase, where copper acts as a activator for this particular enzyme responsible for the formation of brown pigments. It also acts as an activator of lactase enzyme. Now, lactase enzyme is involved in the formation of lignin, where lignin is the structural component of plant cell wall, especially the secondary cell wall, which is responsible for its strength. Copper also facilitates the electron transport reactions by converting it into cuprous and cupric form. Another micronutrient is boron, which is absorbed by the plant in the form of borate ions. It facilitates calcium uptake and also maintains cell membrane integrity during cell elongation and differentiation. It is also involved in pollen germination, and also once the sugar molecules have been synthesized, it helps in the translocation of carbohydrates. Talking about molybdenum, it is absorbed in its ionic form of molybdenum oxide. It acts as an activator of nitrogenase enzyme, which is included in nitrogen metabolism, and also as an activator of nitrate reductase. Both these enzymes play a crucial role in nitrogen assimilation by the plant. So, it is an activator of enzymes which are required in nitrogen metabolism. Now, how is chlorine absorbed and what role does it play in the plant? Chlorine is absorbed in the form of chloride ions, where along with potassium, chloride ions also help in maintaining the ion cation balance by the efflux and influx. It helps in cell osmoregulation and maintaining the turgor. Along with manganese, it is essential for the water splitting reaction in the presence of light during the light reactions of photosynthesis. Welcome back, students. to continue with the last class, today we are going to talk about the deficiency of essential elements. So, how to understand the deficiency of any element? If you see the plot shown on the slide between dry mass yield and the essential elements concentration in the plant, below the critical concentration, which is shown here by the blue color line, to define critical concentration, it is the concentration of the essential element below which the plant growth is retarded. And below this concentration is what is called as the deficiency of the essential element. For actively mobile elements, the deficiency symptoms, they are generally observed in the mature organs of the plant. These include like nitrogen, phosphorus, potassium, magnesium, calcium, sodium, zinc and molybdenum. For relatively immobile elements, the deficiency symptoms are observed in the young developing shoots or tissues, like for calcium, sulphur, iron, boron and copper. So, let us see what are the symptoms which are generally observed when one or more of these essential elements become deficient in the plant. What you see the picture, the yellowing of the leaves, is one of the problems observed, which is called as chlorosis, caused by the deficiency of one or more of the essential elements like nitrogen, potassium, magnesium, sulphur, which are macronutrients, and micronutrients involving iron, manganese, zinc and molybdenum. Another commonly observed symptom of deficiency of essential elements includes necrosis on the leaf tissues, which is caused by the deficiency of macronutrients like calcium, magnesium, copper or potassium. Due to the deficiency of the essential elements, the impact can also be observed in terms of inhibition in cell division. The elements whose deficiency can cause the inhibition on cell division include nitrogen, sulphur, potassium and micronutrients like molybdenum. Delay in flowering is also one of the symptoms of deficiency caused by the deficiency of essential elements like nitrogen, sulphur and molybdenum. Now, how can we avoid the deficiency of these essential elements in plants? It is through application of fertilizers. Now, these fertilizers, they include both the macronutrients like nitrogen, phosphorus, potassium, sulphur and also micronutrients like manganese, iron, zinc and copper. So, these are added to the soil to remove any deficiency of the essential elements in plant. Now, let us see what is toxicity of these essential elements. So, today we are going to see toxicity symptoms caused by the excess of micronutrients present in the plant. So, to understand what is toxicity, again any mineral iron concentration in the tissues that can reduce the dry weight of the tissue by about 10% is considered as toxic. So, if you can see the plot of dry mass yield versus essential element concentration in the plant part, the red region is the toxic region, where if the concentration goes beyond the critical concentration, which is again shown by a blue line after the adequate range, then any excess concentration detrimentally affects the growth of the plant and the plant growth is retarded. So, let us see now some of the toxicity symptoms of micronutrients like manganese. So, when there is toxicity of manganese, one can observe brown spots on the leaves. When manganese is present in toxic limits, then it can act as a competitive inhibitor, where it competes with magnesium and iron and binds with the enzymes and inhibits the activity. Excess of manganese also inhibits calcium translocation in the shoot apex region. Now, let us see what is the mechanism behind the absorption of elements. If you see the picture on the slide, there are two main processes, mechanisms by which these essential elements are absorbed by the plant. One is a passive process, where mineral elements in their ionic form, they will be absorbed by the plant cells in the outer space, which is called as the apoplast region. Another process by which the uptake happens is an active process, in which the uptake of ions happens till the inner space of the cell, which is called as symplast. Now, this is an energy driven process in which these elements in their ionic forms, they pass through the cell membrane, through the ion channels present in the cell membrane with the use of energy. Now, this process of influx and efflux is defined as, so the inward movement of the ions into the cell is defined as influx, and the outer movement of ions is defined as efflux. So, to consolidate, all these mineral elements, they are taken up in their ionic form either through symplastic path or through apoplastic path, where they are translocated through the xylem along with the ascending stream of water, which is pulled up through the plant by the transpiration pull. Now, let us study metabolism of nitrogen. As you all know, air consists of 78% of nitrogen and it is a diatomic molecule. However, plants cannot absorb nitrogen directly from the air. Thus, they have devised a process by which nitrogen is assimilated in its ionic forms in the form of ammonium ions, nitrite ions or nitrate ions by the root system. So, how does that happen? This happens through nitrogen cycle, in which the atmospheric nitrogen fixation happens either through lightning or UV radiation. There is biological nitrogen fixation, in which microbes act as nitrogen fixers, and there is industrial nitrogen fixation via the burning of the fossil fuel. All these processes independently fix atmospheric nitrogen into and convert it into ammonium ions. This process is called as ammonification. Now, the ammonium ion is further through the process of nitrification is converted to nitrite ions and to nitrate ions. And then the plant root system can readily absorb the ammonium, the nitrite and the nitrate form of nitrogen. This nitrate form, the nitrate form can further be converted into atmospheric nitrogen, thereby completing the nitrogen cycle happen. This process of conversion of nitrate back to nitrogen is called as denitrification. Now, let us try to understand what facilitates the fixing of ammonium and converting it to nitrite and nitrates in the soil. So, understanding the process of nitrification. In the process of nitrification, ammonium ion is converted to nitrite ions and further to nitrate ions with the help of microbes belonging to the class of chemoautotrophs, where conversion of ammonium ions to nitrite ions takes place, for example, by the species of Nitromonas or Nitrococcus, and further nitrite ions can be converted to nitrate by the Nitrobacter species. The process of denitrification, this is carried out by some of the bacteria from the family of Pseudomonas or Thiobacillus. The atmospheric nitrogen is fixed to ammonium by the process of biological nitrogen fixation, which is carried out by nitrogen fixers. These are free living organisms in the soil, can be aerobic or anaerobic, like for example, Rhodospirillum, anaerobic class of bacterium, and aerobic organisms belonging to Azotobacter species, or for example, cyanobacteria. All these species help in the conversion of atmospheric nitrogen to ammonium ion. So, in the biological nitrogen fixation, it is the symbiotic relationship of these organisms with the plant which is exploited, which works in fixing the atmospheric nitrogen to ammonium ions for the plants. Nodule formation is seen in the plant roots. As you can see the picture on the slide of the Rhizobium species, where the Rhizobium, it resides in the roots of the leguminous plants. You can see the microscopic picture of the root section. For example, the plants belonging to broad beans, peas family, lentils. So, there one can observe nodule formation in their root systems. There is another example of nodule formation where Frankia species, these are filamentous bacterium which can form in non-leguminous plants the nodules and act as nitrogen fixers. For example, the plant like Alnus. Now, how do these root nodules develop? Here is an example shown for the Rhizobium species. How do they act on soybean roots and they form the nodules. So, these are the different steps of its progression. The bacteria, it grows around the root hairs, and once it colonizes the root hairs, it travels like a thread into the inner cortex and pericycle cells which are under division, and then the root hair curls up like a hook and it forms a mature root nodule. This is a picture of the soybean root nodules. So, the symbiosis is that the soybean plant, it provides the carbohydrate supply to the rhizobium, while the rhizobium helps the plant in fixing the atmospheric nitrogen. So, the mechanism involves, it is an energy driven process in which the enzyme nitrogenase is used, where the atmospheric nitrogen is biochemically converted to ammonium ions. Now, this reaction is sensitive to oxygenase. So, the nitrogenase enzyme being sensitive to oxygen, it is protected by a molecule which is called as leg hemoglobin, so as to protect it against oxygen. This pink coloration of the nodules section, if you can see from inside, it is pinkish in color. This color is because of the leg hemoglobin present, which acts as an oxygen scavenger. So, now let us see what is the fate of ammonia. At physiological pH, ammonia is exists in the form of ammonium ions, and ammonium ions in excess can be toxic to the plant. So, plants have devised a way to readily absorb ammonium ions and convert them to synthesize amino acids and use them to synthesize amino acids via reductive amination and transamination reactions. In the reductive amination, the ammonium ions, they combine with alpha glutaric acid and NADPH. This reaction is carried out via glutamase dehydrogenase to obtain L-glutamate, while in a transamination reaction, your amino acid combines with keto acid in the presence of transaminase. So, this reaction is carried out by transaminase, converting it into another amino acid. So, to summarize the chapter on mineral nutrition, we defined mineral nutrition and mineral elements. We classified the elements into essential and non-essential elements based on their quantitative requirements in the plant. The essential elements were further classified into macronutrients and micronutrients, where macronutrients, they constitute structural components of biomolecules, and micronutrients, they function as activator or inhibitor of enzymes. Based on critical concentration of mineral nutrients, they can be either deficient or toxic to the plant. Mechanism behind absorption of elements includes active process as well as passive process. And then we went on to study the nitrogen metabolism, the nitrogen cycle, how atmospheric nitrogen is converted into ionic forms for assimilation by the plants, and then the ionic form is reconverted by the process of denitrification into atmospheric nitrogen. Then in the end, we also saw briefly the fate of ammonium ions.